to turn off your camera. Sneha's going to go ahead and start rolling the camera. Oh, look, she started. So I'm going to start. My name is Sarah Kick. I am a senior scientist at the Estuary Partnership. I've been working in these watersheds since 2008. I'm super excited to talk about water quality today with everybody. I want to go ahead and pass it around and have everybody introduce themselves because we're a pretty small group. So I'm going to pass it to Sneha.
Hi, everyone. I'm Sneha. I am the research scientist at LSIP, and I'm going to be working with Sarah on presenting some of the results here today, and I'm very excited to be part of this group.
Celine, do you want to go next?
Sure. For those of you that don't know me, my name is Celine Keeney. I'm with the Columbia Soil and Water Conservation District. I'm a senior resource conservationist, and I'm basically the project manager of the water quality on the SWCD side for the water quality monitoring program.
Sweet. Dana?
Yeah. Hi, I'm Dana Pritchard. I am the coordinator for the Scappoose Bay Watershed Council based out of Warren.
Jenny, I'm just calling people because this is the order I see in my screen. So I'll just call everybody out.
Hi guys, I'm Jenny Dezo from the Lower Columbia Estuary Partnership, and I'm a restoration ecologist. And I love the Scappoose and CLATSKANIE watersheds. So thanks everybody for helping protect these areas. I'm excited to work here.
Ian?
Hi, I'm Ian. I'm an intern with LSIP. Excited.
Welcome. Alan, you're next on my screen.
Alan Whiting with the Lower Columbia River Watershed. I'm also pretty excited to see some of the data come out, so happy to be here. Thank you.
Katie?
Hi, I'm Katie Duzick, and I'm with the Watershed Enhancement Board. I am the regional program representative for the North and Central Coast regions, which includes the geography of this monitoring effort.
Awesome. Elaine?
Hey everybody, I'm Elaine Placido, the Executive Director of the Lower Columbia Estuary Partnership, who can obviously not speak today very well. I'm happy to see all these faces on here and to be here to support my amazing team present their work.
Oh, I got a few folks without their video on. I'm just going to call your name, and if you want, you can introduce yourself. Dave?
I'm guessing that's me. And my name is Dave Suka. I'm with the City of Scappoose Public Works Department.
Welcome. Daniel? Oh, Daniel wrote a chat. Let's see if I can pull it up. Oh, wait, no, that.
Yeah, Daniel is from Columbia County Public Health, Daniel Osborne, and he doesn't have his mic or his camera set up. So I'm going to monitor the chat for you guys, and I'll let you know if there's any questions or comments. So I invite everybody to use that chat if it's helpful.
Cool. Thank you, Jenny, for doing that. I think I see Jim.
Hi, I joined a little bit late. It looks like we're doing introductions. I'm Jim Brick. I'm the fisheries biologist with the Oregon Department of Fish and Wildlife located in Clackamas.
And thank you. And let's see, Aaron.
Hi, I'm Erin O'Connell with Columbia County Public Health. I just wanted to listen in on the findings because I'm pretty interested in what you guys found from a water quality standpoint. I do all the septic systems, so kind of, yeah, eek. Thanks.
Thanks. Now does anybody see more folks? I'm not seeing any more on my list. Okay, that's everybody. Well, thank you everyone for your introductions. So I'm going to go ahead and share my intro slide, I think. And then I'm going to let Celine take over with an introduction. Or maybe I could let Celine take over. Aha. All right, Celine, it's all yours.
Okay.
If people could mute their mics, while we're presenting, it'd be really helpful. Thank you.
So before the presentation gets kicked off in earnest, I just wanted to provide some background information. So in 2017, the Columbia Soil and Water Conservation District established this water quality monitoring program in collaboration with the Lower Columbia Estuary Partnership to track temperature, turbidity, and E. coli bacteria in the CLATSKANIE River and Beaver Creek, which are two major tributaries to the Lower Columbia River, and in Milton Creek, North Scappoose Creek, and South Scappoose Creek, which are major tributaries to Scappoose Bay, which flows into the Columbia River. So the SWCD is the program sponsor and provides partial funding for the program as well. And the Lower Columbia Estuary Partnership collects the water quality monitoring data and reports on it. The other project partners include the Oregon Department of Environmental Quality, DEQ, and the Oregon Watershed Enhancement Board, OWEB. DEQ grant funding facilitated the establishment of the program in 2017, and OWEB grant funding has facilitated continuation of the program since 2017. So in this presentation details the results of the monitoring conducted from 2017 to 2020. But as a side note, there was water quality monitoring conducted in the Scappoose Bay tributaries from 2008 to 2011. So that data set has been incorporated into the report that the water quality monitoring results report completed by the Lower Columbia Estuary Partnership and detailed in this presentation. So for those of you on this virtual meeting, and then if you would like to refer to any of this future information, it will be posted to the Columbia SWCD website in addition to the recording of this presentation. So just look for it in the coming days if you would like to pass along what's being presented today. So with that, Sarah, you want to take it away?
I'll take it away. Thank you, Celine. That was a good background. So we're just going to ask that folks hold questions until the end because we want to be really aware. We're really aware of everyone's time today. And so Sneha and I are going to tag team a presentation. It's going to be about 30 minutes long. We are going to try to break down some of that really heavy data that we put in the report. If anyone has had a look at the report yet, you'll know what I mean, into something really accessible. And then after the presentation, we're going to open it up for any questions you might have. And then of course, like discussion about next steps. So we're really excited to do that with everybody. I'm going to try to change my slides and make sure it works. So we have just talked a little bit about the history of the monitoring program. I'm going to talk a little bit more about it. Sneha is going to give us some really good descriptions of the watersheds that we're monitoring. We're going to go into our monitoring methods and the monitoring frequency that we've been doing. Then we're going to talk about the results, and we're going to really focus today on the temperature and the E. coli. We're going to touch briefly on turbidity and conductivity, but really we're focusing on the temperature and E. coli results because those are the ones that really popped in the report in terms of things to make note of. We're going to have some summaries and then, of course, the next step. So we're really excited to jump into this today. This is a picture of Lower CLATSKANIE looking upstream. So you can see that's really pretty. We just took that the other day. So Celine did a great job kind of going over this program history. I went ahead and just kept this slide in because we had planned on talking about it. I do want to talk a little bit more about that 2008 to 2011 study. I was really lucky to be participating. I was an employee of the Watershed Council at the time, and I had my hand on all that data at the time. And so it was really like serendipitous when I was hired on at the Estuary Partnership. And my first week, I got to go out with Celine and talk about water quality monitoring. So it felt like it was meant to be. And so I've been really thankful to be continued to be a part of that program and to talk about that data today. And we're just really excited to take these data, continue collecting them. We really want to do that 5-year plan of collecting the data. This is just a report out during this monitoring period. We want to take comments on how the data is being analyzed, continue to update our approaches, and use that to inform restoration management, and of course, long-term monitoring. Because the idea here is that we are going to start making management changes and we're going to start making restoration changes. They're going to impact these watersheds. And we want to be able to detect those changes, those positive changes in water quality. And so keeping a long-term track of this water quality conditions is really critical. So I've already touched on this, but I will talk a little bit more about the fact that for the 2017 to current water quality monitoring program that we talk about in the report, we have expanded monitoring beyond SCAPPOOSE watershed sub-basins into CLATSKANIE and Beaver Creek. And so across all of these, we have 13 different monitoring locations. And we, in this most recent report, did a landscape cover analysis to accompany the water quality analysis. And in the 2008 to 2011 report, that data, we looked at the land cover from 2001, because land cover takes a long time to come out. So right now, the most current land cover you can get your hands on is 2011. Sneha did an analysis to compare the 2001 and 2011, and she's going to go ahead and start talking about that in the next slides. So I'm going to go ahead and pass it back to Sneha.
Thanks, Sarah. If you don't mind controlling the slides for me, that would be awesome as well. Do you mind going on to the next slide? Thanks. So the first watershed we'll take a look at is the CLATSKANIE River watershed. The watershed has an area of nearly 47,000 acres, and it drains into the CLATSKANIE River, which is 26 miles long. According to the land cover analysis that we conducted from USGS data of 2011, we found the watershed is mostly forested. It has about 4.5% of pastures or agricultural land, and about 4% of it is developed. Across this watershed, we sampled at 5 locations. 3 of these locations are on the main stem, and those are Upper CLATSKANIE, Middle CLATSKANIE, and Lower CLATSKANIE. We also sampled 2 tributaries, that is the Carcass Creek and the Little CLATSKANIE. In terms of land cover, Upper CLATSKANIE and Little CLATSKANIE are in the more forested areas of the watershed, whereas Carcass, Middle, and Lower are all in the pasturous areas. Lower and Middle are towards the more developed areas as well. Next slide, please. Moving on to the Beaver Creek watershed, which is adjacent to the CLATSKANIE River watershed. This watershed has an area of about 31,000 acres. This one is also mostly forested, with about 11% as pasture or agricultural land, and it has a little over 8% as developed land. We sampled across 2 sites, and in this watershed, the first one was Upper Beaver, and it's present in the upper area of the watershed, and Lower Beaver, which is towards the lower areas of the watershed. Both of these sampling sites are present in locations that are more residential and more pastoral in nature. Next slide, please. Moving on to Milton Creek watershed, which is, as Sarah said, part of the Scappoose Bay larger watershed. This one has an area of about 20,000 acres and has 13% of pasture and agricultural land, and 9% of it is developed. We have our current monitoring sites depicted as pink dots on this map. The first one is Upper Milton, and this is present towards the upper areas of the watershed, which are more residential and pasturous, whereas Lower Milton is present near the developed area of the watershed, and it's close to a public park. I have also depicted the locations of the previous 2008 to 2011 monitoring efforts through green dots. And as you can see, there are a couple of locations in the upper watershed and 1 location in the lower watershed. Next slide. The North Scappoose watershed has an area similar to Milton Creek, that's about 20,000 acres, and this is largely forested. It has 6% of land that's either pasturous or agricultural land, and 6% that's developed. It drains into the North Scappoose Creek, which is 12 miles long. Again, on this map, I've denoted the locations for the current monitoring efforts as pink dots, and one of them is in the upper North Scappoose, that's in the upper watershed. It's more in the residential and forested area, whereas the lower North Scappoose is present towards the lower watershed, which is more developed. We also compared our results to the 2008 effort, and the sampling locations from that effort are depicted as green dots on this map. Next slide, please. Last but not the least is the South Scappoose watershed. This one is smaller in area, just covers about 17,000 acres. It has 9% of its land that's developed and 7% as pastures. This one drains into the South Scappoose Creek, which is again another 12 miles long. We have 2 sampling locations as part of our current effort depicted as pink dots on this map. The first one is Upper South Scappoose. It's also present in a residential forested location. And the Lower South Scappoose is in the lower, more developed area of the watershed. Locations that coincide with the 2008 to 2011 monitoring effort has been depicted through the green dots. Next slide. Now that I've given a rundown of the watersheds, I'd like to talk a little bit about our metrics. And as you can see, this is an image of a lot of people deploying data loggers for our continuous temperature monitoring. And the one thing we learned from this effort was not to tie a data logger to a tree, even though it looks stable, because then you're going to have storms and the tree is going to get washed out. But moving on, we collected a suite of parameters in this program. We collected E. coli data, temperature, conductivity, and turbidity. And as of 2021, we've also included pH and dissolved oxygen. We curated a long-term temperature data set from 2017, and it's ongoing by deploying data loggers at all 13 monitoring stations. We also collect turbidity samples on a monthly basis at all of these locations. We collect on-site, in situ, instantaneous temperature and conductivity measurements at these sites. Our E. coli sampling has gone over more of an evolutionary trend. We started with collecting monthly samples during the summer at just some of the sites, and we expanded it based on the preliminary results to cover all year long monthly and bi-monthly sampling at all of these locations from the fall of 2019. Next slide. Now, we also collect our water quality parameters in accordance with protocols issued by the DEQ. We also have a sampling and analysis plan that we developed with the DEQ, and we're more than happy to share that with anyone who's interested. This slide basically shows the parameters, the equipment that we use, and the accuracy of those equipment, as well as the acceptable ranges or the thresholds that we have compared our results to that we'll be presenting today. Now, moving back to the E. coli evolutionary trend, the reason we changed the way we monitored these sites or sampled at these sites was so that we could compare our results to the DEQ regulatory limit of a 90-day geometric mean of 126 E. coli organisms per 100 ml. And we need a minimum of 5 samples within those 90 days. And no single sample can exceed 406 counts per 100 ml. And with this, I think I'm going to pass it back to Sarah, who can take us over some of the results.
Sorry, I had to unmute myself there. Thank you, Sneha, for that. All right, who's ready to get into the results? So I don't know how many of you had a chance to open the report. We heavily relied on box and whisker plots to show the range in data. And I feel like these are really useful. However, if you've never seen one before, you may think they're very confusing. So I just wanted to point out how to read one. Essentially, what you're looking for is that the most of your data should fall in the middle box. So essentially, the whiskers are the limits of your data. Most of your data is going to fall, or almost 100% of your data is going to fall between the two whiskers, and then that box in the middle is 50% of your data. It won't let me use my mouse for some reason to point that out, but I know that you can see it on there. So I'm going to be showing a bunch of these today. If it's very confusing, please feel free to interrupt me. But I think that we have tried to, in the way that we're presenting the results today, I don't want anyone to get overwhelmed by the graphs, because we have tried to include things that are not graphs that also convey the results. So just hang tight, and I promise it'll make you happy one way or the other. Okay, so Sneha's making faces. Okay, so talking about temperature. So the way that we're going to talk about the data is that we're going to first talk about one parameter, and we're going to go through all the results for all the different watersheds for each parameter. So this parameter is temperature. Temperature is really important. It's really important because it really dictates the habitat conditions for these animals, the organisms in the stream and the fish. If you have really hot water quality, nothing can really survive in that water. It also is going to very, very high temperatures. It's going to cause an increase in algal blooms, in addition to easier conditions for bacteria to grow. So high water temperatures are not good for habitat conditions. Today, we're going to primarily be talking about thresholds for salmonids. That's something that was really commonly used when talking about water quality. And 18 degrees Celsius is the primary threshold we're going to discuss. If a site is over 18 degrees Celsius, it's considered outside of the ideal range for salmonids. And so if it's outside of 18 degrees C, we're going to call it out in our data. You're going to see it in our graphs, and we're really using that as our primary threshold. Now, over 25 degrees Celsius is also considered lethal, so that's something to keep in mind as you're looking at the graphs. I'm also going to use this temperature icon. If the temperature icon is red, like you see here, that means that the water is too hot and we are breaking that threshold during the summer months. So I'm going to flip the results on their head. I'm going to tell you the summary of all the data right now. So we saw occurrences across all basins of greater than 18 degrees Celsius. The most ideal cool water temperatures were primarily found in the upper basins, the upper portion of the watersheds, which is to be expected because most of these are very forested. The water also experiences thermal loading as it's moving through these watersheds. So you do expect there to be some increase as the water moves down into the lower basin. Now, something that was a little striking to us and was very clear was that there was a temperature increase between our current data set and that 2008 to 2011 data set. We saw a significant increase in water temperatures, especially in those lower reaches. And so I'm going to start talking about this in detail now. So the first site we're going to discuss is CLATSKANIE, the CLATSKANIE watershed. Now, the first thing that you see, or I'm going to direct your eyes to that box and whisker plot. What that is depicting is the temperature on the y-axis and the months on the x. So you can see that the monthly average water temperature increases over the year, as you would expect, with the highest temperatures in August. Now, this is true for all of our watersheds. Now, if you look at the map, I'm going to show you that those upper reaches of Little CLATSKANIE, Upper CLATSKANIE, and Carcass Creek are staying really nice and cool in August. This is an August temperature map. And so we see that those are staying below the 18-degree threshold. However, we do have occurrences of above 18 degrees for Middle CLATSKANIE and Lower CLATSKANIE. And so another way to look at this is to look at some beautiful pictures of these creeks. Now, if you look at these pictures, you can't tell whether the water is hot or not. You know, these all are very beautiful, but I put those little temperature icons on there for you. You can see that we have some issues with temperature in the Middle CLATSKANIE, and it's pretty hot in the summer in Lower CLATSKANIE. However, those upper reaches are staying really nice and cool. Do people hear a lot of feedback from me right now? I hear some feedback from myself. Okay, good. Okay, so the same map, but now we're looking at Beaver Creek. I've included CLATSKANIE because it's adjacent to Beaver Creek watershed, what you'll see here is that overall, lower Beaver and upper Beaver are showing to be a little bit warmer in August. Those summer months, they heat up. And especially that upper Beaver Creek is pretty warm compared to those upper reaches in CLATSKANIE. And so very similar to what I showed you in the pictures, you can see that upper Beaver has some questionable temperatures. About half the summer, it's crossing that threshold. Lower Beaver, it's even more of that time period. It's crossing that 18-degree threshold in the summer. Okay, now I'm going to change how we're seeing the data. Don't get mad. I took away the box and whisker plots, and now we're looking at a table. It's okay, I promise. So what you're going to see is on the map, I'm going to direct your attention to the map. We have mapped out the historic data and the current data so that everyone can have a really good view of what those data look like, especially for August here. And then in the table above the map, I have the monthly average temperatures across the months of the year. And you can see the comparison between 2008 to 2011 and 2017 to 2020. The most important thing to point out here is that for the upper basin, mostly historically it stayed below 18 degrees Celsius for the summer months of July and August. However, what we observed in the current data set is that it was actually staying above 18 degrees Celsius for the summer months. Now, our monitoring location is a little bit lower in the watershed. However, I do think that it is a significantly warmer. So it's something to consider as we move forward. Lower Milton, it was pretty hot in 2008 and 2011. It remained above 18 degrees Celsius for July and August. Now we're seeing it stay above 20 degrees Celsius. So it's really warm down there at Lower Milton for the 2017 to 2020 data period. Again, these are the summer months. Okay, so looking at those pictures again, Upper Milton has some temperature issues. Lower Milton, you don't want to be a salmon in Lower Milton in the summer. Okay, North SCAPPOOSE Creek. This is looking very similar to Milton. We have that historic data set we're trying to present here. I want to point your eyes to the map. You can see in the upper basin, our 2017 to 2020 data, it's above 18 degrees Celsius for about half of July and August. Historically, it was pretty much below that. Now, I do believe that the historic data set was collecting an Alder Creek sub-basin, but again, these sites are very close to each other in the upper basin, so I do think it's important to note that difference in temperature. The lower basin, very close to each other. Very similar to Milton Creek, we have above 18 degrees Celsius historically, but now we see it above 19 degrees Celsius for those summer months. So again, a significant increase in temperature. Some pretty pictures. It's pretty warm out there, even if it looks nice and cool. All right, South SCAPPOOSE Creek, using that same platform to show the historic data. I think it's important to note, it looks very similar to those other stories I talked about where in the lower basin, we see warming. However, good news, upper basin stayed cool. So our historic data set and our current data set shows that the upper basin is staying below 18 degrees Celsius. That's a win. Okay, good. All right, moving on to the picture slide so we can look at that. Oops, sorry, I admitted Celine and now it's not letting me change slides. We have our glorious field team out in these pictures. They think the water is very cold in upper South SCAPPOOSE Creek in the middle of summer. Yay! But lower South SCAPPOOSE Creek's warming up in the summer, warming up. All right, now I want to point, Sneha is in Milton Creek right now, looking down, making a determination. Should I put my hand in that water? Sneha's going to talk about E. coli in the watersheds.
Thanks, Sarah. Well, while I was thinking about whether or not I had to put my hands in, I decided I should because, you know, we need all the data we can get. But anyway, let's start with what exactly E. coli is. So it's firstly a form of fecal bacteria which comes from human and animal waste. And generally, its high levels of E. coli is considered poor quality for the stream, and it poses health threats to humans as well as salmonids. I would like to remind everyone that we will be comparing our results to the EQ regulatory limits of the 90-day geometric mean of 126 E. coli counts per 100 ml and the no single sample over 406 E. coli counts per 100 ml as well. Before we take a look at the whole, at each, like the, before we take a look at the more detailed results, I'd like to talk about a few highlights, and that is basically that we have high levels of E. coli in the summer and the fall at one of the locations in the CLATSKANIE watershed and several other locations as we move down away from the CLATSKANIE watershed. Let's go into the more detailed results. So firstly, let's look at more box and whisker plots and maps. So this one shows basically the trend of how E. coli moves across the months of the year. As you can see, most of our sites, the E. coli stay pretty low in winter and spring months, and then around June, these E. coli counts start elevating, and we see these numbers stay pretty high all the way till November. Middle CLATSKANIE, though, is the exception because it stays higher than all these sites throughout the year, and it exceeds the 126 DEQ 90-day threshold of 126, and it exceeded this between the summer and fall of 2019 as well as 2020. Here on the map, we have depicted where the locations of these sites are and the highest or the maximum 90-day geometric mean for the entire data set. And as you can see, the upper areas of the watershed, they're pretty low. And then when we move closer to the more residential, the more developed areas, we see that lower CLATSKANIE also, it's getting close to the threshold, but middle CLATSKANIE in August, as of August of 2020, it has already crossed the DEQ 90-day threshold. Next slide, please. So if you were deciding on going swimming, I would suggest staying in the upper areas of the watershed because the lower areas, not so good. Moving on. Next, we move to the adjacent Beaver Creek watershed, and we've placed the CLATSKANIE numbers also on the map for comparison. We see significantly high 90-day geometric means in the upper Beaver location, and the lower Beaver, technically it stays below the geometric means, but it does have instances where the single sample limit does cross the DEQ regulatory standards. Next slide, please. So as a summary, upper Beaver, it does have some E. coli issues that we need to take a look at and focus our study on. Moving on to the larger Scappoose Bay watershed, Milton Creek is one of the first watersheds that we would take a look at, and between summer and fall, the 90-day geometric means for our current monitoring effort, that's the upper Milton and the lower Milton, both of these exceed DEQ limits of 125, 126 mpn per 100 ml. Lower CLATSKANIE, though, it exceeds 200 E. coli counts per 100 ml as its geometric mean. When we compared these results, when we compared overall means of the previous historic results to our current monitoring efforts, we have seen that E. coli has been a persistent issue in this watershed. Next slide. So in a more visually pleasing way, in the summer, we definitely need to take a closer look at the E. coli issues of this watershed. Moving on to North Scappoose watershed, we see, again, similar to Milton Creek, higher E. coli counts in the lower areas of the watershed in the summer months. And similarly, the upper areas of the watershed, they are high, but not as high as the lower, more developed areas. When we compared the overall means of the current effort to the previous effort, we did see that E. coli levels have been steadily increasing. Similarly, in the summer, maybe you should go to CLATSKANIE watershed to take a swim. Moving on. Lastly, it's the South Scappoose watershed, and this one also follows similar trends to the Milton Creek and North Scappoose Creek watershed. The lower areas of the watershed have E. coli levels that are higher than the DEQ limits. The only thing is that here we couldn't compare the overall means of the upper watershed because we didn't have enough samples to perform that analysis. However, in the lower watershed, we have seen that the E. coli levels have steadily been increasing. And this is our beautiful team wondering whether they should really collect the sample at lower South Scappoose. All right, Sarah, this is back to you.
All right, thank you. Thank you for that stimulating assessment of don't go swimming. Okay, so for turbidity, I know that folks on the call are really interested in the turbidity results, and so I am going to briefly touch on them. Turbidity is essentially referring to the clarity of the water, the amount of suspended solids in the water. We don't directly monitor the total suspended solids, but turbidity is a proxy for that. We do know that over 10 NTU is a threshold for aquatic organisms. Too much turbidity essentially covers the stream channel in debris. It really limits macroinvertebrates, and it's very poor water quality. Very, very high turbidity levels can essentially kill fish as well. So something to keep in mind as we go through these results. Oh, sorry, here, let me. A summary of our overall results for turbidity is that we did observe, although we're just doing monthly grab samples of turbidity, we did observe events over 10 NTU across most of the watersheds except CLATSKANIE. The levels were really variable, however, and they didn't necessarily have a seasonal trend. It was varied by watershed. We did notice that the historic conditions were higher. The turbidity levels were higher in 2008 to 2011 than they are currently across the Scappoose Bay watersheds, which was kind of a nice thing to find that our water quality in some ways might be getting better. So jumping into these data, for CLATSKANIE, we did see variability in turbidity levels. Higher turbidity levels were observed in that Little CLATSKANIE and Upper CLATSKANIE watersheds. The lowest turbidity levels were found at Carcass Creek subbasin. However, there were no occurrences that we observed of over 10 NTU. And so generally very low turbidity levels. Now for Beaver Creek watershed, again, these graphs look very similar to the ones that you've already seen where we have showing lower Beaver and upper Beaver relative to lower CLATSKANIE and upper CLATSKANIE areas. Upper Beaver did have occurrences of over 10 NTU and generally had a very high average of 7 NTU, 7.1. So that's something to kind of keep our eye on for upper Beaver. And again, upper Beaver, unlike lower Beaver, is a little bit more developed, more pasture lands up there in that subbasin. And then essentially this watershed becomes more forested. So it's a little up on its head in terms of upper Beaver versus lower Beaver Creek. Now for Milton, now we're looking at the historic data. So you're probably looking at this box and whisker plot and going, oh my goodness. Well, if you don't like box and whisker plots and that looks like too much data, just look at the map. And what I want to point out here is that the averages in Milton Creek are around 5 to 8, and that's including the historic range. And what we did see is that the current data set, that 2017 to 2020 data set, generally our averages were just a little bit lower. So we do see less occurrences of over 10 NTU, and then the overall averages were lower for those levels. However, for the Scappoose Bay watershed, Milton Creek does have the highest averages still. So as we go through, you'll see that. North Scappoose Creek, generally very, very low, historically low and currently low. No real changes, maybe a slight decrease in overall turbidity levels between the two data sets. But again, fairly good, very low occurrences of over 10 NTU that we observed. And then for South Scappoose Creek, very similar story, fairly low levels, very few events of over 10 NTU, especially when compared to the Upper Scappoose historic data set. Now I want to talk very briefly about conductivity. We reported out on it just like all of the parameters. It's not something that we it's something that's really good to monitor as a baseline. Conductivity as a measure of electrical current in the water, it's used as a proxy for pollution. So developing a baseline understanding of what your watershed's conductivity is can be very useful for long-term monitoring, because if you are going to see an increase in pollution, you're going to see a change in your conductivity, or if, let's say, you reduce stormwater runoff, you might see a decrease in conductivity. So it's a really good monitoring metric to keep an eye on those trends over time. The one thing that I will point out is that so this box and whisker plot that I just put up there has us a hatched line at 150. Typically, watersheds in the Pacific Northwest have conductivity below 150 microsiemens. Historically, the data set was well below 150. In South Scappoose Creek, we did see occurrences of over 150 microsiemens in that lower monitoring area. And my guess is that has to come from storm events and stormwater management. So, brake pads, water pollution, cars, those are all things that are going to increase conductivity. So that might be something to keep an eye on, but it wasn't a red flag. I just wanted to mention it. I'm going to pass it back to Sneha, and she's going to do some interpretation of these results for everybody.
Thank you, Sarah. Now, obviously, this has been a large data dump on the audience, but I'd just like to show prettier pictures and give like a general summary of the main water quality issues that we observed through our results. So first, let's look at the CLATSKANIE watershed. Over here, most of the water quality issues were isolated towards the lower reaches of the watershed. That means at middle CLATSKANIE and lower CLATSKANIE. We had high summer temperatures that exceeded DEQ thresholds. Same case with bacteria as well. During the summer and the fall of 2019 and 2020, we saw elevated levels. Turbidity, not so, we didn't see a lot of turbidity issues in this watershed. Just seasonably high whenever there was any high storm event, but nothing that was persistent. Beaver Creek watershed is, well, in terms of temperature, in terms of summertime temperature, we need to keep an eye on both of these locations. It's in terms of E. coli and turbidity, Upper Beaver definitely needs to be taken into consideration. We need to, do some restoration or maybe do a more focused study on what exactly is causing some of these issues. According to our results, what we do think may be happening is that since Upper Beaver is in the more agricultural, the more pastoral area, you may have some excess runoff or something like that that's happening. Whereas the temperature issues in Lower Beaver Creek, we think is an issue because of thermal loading as the water moves down the watershed. Lastly is the larger Scappoose Bay watershed. We've clubbed the results of these three sub-watersheds because the trends are pretty much similar in nature. We see high temperature as well as E. coli levels in the lower areas of the watershed. Turbidity, not so much of an issue in the North Scappoose and the South Scappoose, but in comparison, Milton definitely has a little bit of a higher turbidity issue according to our results. So, and based on all of our results, I think we have suggested a few different types of studies and restoration actions in the report that Sarah will talk about and conclude it.
Thanks, Sneha. So I know that we've been talking to everybody for about 35 minutes, so thank you for listening. We wanted to just talk briefly about next steps and then open it up for questions and discussion. We are currently collecting dissolved oxygen data right now. We've added that to our monitoring and pH. These are grab samples at the time of data collection. So that is monthly in the fall through spring and then bi-monthly in the summer. we'd like to plan to continue doing this monitoring through 2022. We have a pending OWEB grant to do this work, and we have a few more reports coming out. So we want to take your feedback, think about how we're analyzing this data, think about maybe focusing in on a sub-basin, looking specifically at the drainage area for each monitoring location. What does that land use look like? Maybe we can do a canopy cover analysis, isolate some trouble areas, and really identify sources of pollution and restoration and management opportunities. The potential to do a bacteria focus study to identify the sources of bacteria might be very useful, thinking about agricultural runoff or potentially septic leakage. These are all possibilities. I know that there's a lot of interest in emerging contaminants, and I think the Oregon DEQ is working on some of that, and the Fish and Wildlife Department. So I think these are all things that could be integrated into a larger, longer study of these watersheds. They definitely deserve it. And so when we move forward, management and restoration recommendations right now, we really can't stress this enough. We think that there needs to be public signs to warn people about high E. coli in those public access areas. Sneha and I and the team have been out to these areas all year long. We see people in the water. We see people playing in the water. We worry about them. We worry about ourselves. It's something that is serious. We need to take this seriously. Things that we can do right now, thinking about stormwater management, riparian buffer management, these things can really help reduce runoff and really reduce a lot of these water quality monitoring issues. And then really focusing on the Scappoose Bay watersheds and the Beaver Creek watersheds for those suggestions. So I wanted to point out in this slide that we have both Chris and Celine. I know Chris isn't on the call today, but Celine, there you are. Look at that, out there with us, water quality monitoring. So I got a photo of you in there. Also, it's a time for me to say, hey, thanks Jenny Dezo and Matthew Schwartz for all your help along the way collecting data out in these watersheds. So, and I'm sure I missed a lot of folks, but I wanted to thank everybody, and we are now happy to answer any questions and then really get the discussion going. Is Celine still on the call? I saw she got kicked off earlier.
I'm here.
Okay.
I'm back.
Well, I can stop sharing so we can see each other here. Oh, we did such a good job. Nobody has any questions. Any questions for yourself?
I have a question. This is Dana.
Oh, hi, Dana.
So you mentioned potentially looking at E. coli sources. So I'm assuming you're talking about doing some genetic analysis of the E. coli to figure out if it's from humans or animals or what. What does that entail as far as additional costs or additional monitoring needs to get that information?
Gosh, that is a good question. We have not looked into exactly what that study would entail, but we have identified it as something that we want to do. So I think that's a good point. I hadn't, Sneha, did you have anything you want to add to that?
I mean, the point of E. coli bacteria, even the amount that we are sampling right now, according to me, it's less. If you want to do a focus study, we have to, because we go out once a month and maybe in the summer, not maybe, in the summer twice a month. And that it's in that result itself, we see variability. So to reduce that variability, you need to have more samples, and then maybe a subset of those samples you can definitely apply the DNA genetics to, and then that could be your focus. The other type of E. coli investigation you could do is based on like the GIS sub-basin analysis, basically seeing what are the sources that are entering into that particular sampling point. These are some of the ways that you can actually determine what sort of E. coli is entering into your streams and, target your restoration actions towards those.
Yeah, I will add that at one point, I think it was in 2018, we tried to kind of do like a mini study where we did a pulse where we went and took a bunch of samples across different locations to see if we kind of like isolate where it's at. But what we ran into was that idea of that variability. So like, it can change very quickly, and without consistent monitoring over a short time period, you're not going to be able to isolate it that way. And the trick with the E. coli, it is our most tricky and expensive thing to monitor because we have a very short period of time that we can hold those samples before we have to get them to the lab, and then the lab has to analyze them very quickly. So the hold time is an issue. And so not to say that I think that something that Sneha and I would like to work with others in terms of coming up with a good plan and maybe a focused area to start a study. I think that we probably would not be able to attempt it across all watersheds at once. It would be something where we'd have to pick one site and be like, okay, let's narrow in on this one. I know other folks have expertise on the call. Does anybody want to chime in about this? Aaron?
Yeah, I would like to, because these were results that aren't really surprising, and I kind of expected to see this, but definitely I think that, so for Milton Creek, for example, where you guys were seeing some really high numbers, I mean, we have a whole area, a development there, the Furlock Park area, that they're all older properties, they're all older septic systems. It's not currently sewered, and I think I would like to find time to sit down with you guys at some point and be part of this process, because I think not only is this important from what I do from a septic perspective and trying to get people to, repair their septic systems appropriately, but also in this particular location, I think this could be really useful information for making the point to the city of St. Helens about elevating a plan to sewer that area. I mean, that could be huge from a water quality standpoint, because I mean, we're talking, I don't even know how many houses, but definitely over 100 in that small area. And if they're all on septic and they're all old, I know that's extremely, that is contributing to this problem. It would not surprise me one bit. And I think we can just start to make, like infrastructure plans and that sort of thing that can be a part of this, in addition to the restoration, right? Because you guys can come in and do restoration, but if this was really coming from septic systems, then we need to look at this from a little bit higher regulatory level, and how can we clean this up and get the, get the community behind that and the actual jurisdictions behind that to focus efforts.
That is really good information, Aaron. We will definitely reach out and talk to you.
Yeah. Thank you so much.
Yep.
Hey, Aaron, have you guys, this is Jenny from LSUB, have you guys done like a mapping of where the known older septic systems are? Do you have any mapping of that, or?
So I don't have mapping. What you have is this right here, because I've been doing this for 13 years. And so you really get to know your areas, and you really get to know sort of the lay of the land and what you expect. I mean, I think it's really important, like you said, on the bacteria stuff, because we do need to figure, is this human caused, right? Because it could be other animal. And I think that sort of drives this process. And I could probably anecdotally go through all of the areas that you showed of why I think septics could contribute in that particular area. But yeah, no, I don't have a map, but I do think I have enough knowledge just rattling around upstairs that I probably could give us a really good starting point for maybe, what does this look like? Where should we focus? How can we carry this forward? I'm a super busy person, but I think this is really important from a public health and water quality standpoint.
Yeah, that might be great to sit down with a map about that, because I was just kind of thinking of those high E. coli areas. One of them was Beaver, where it sounds like there's a lot of agriculture already, so we might be able to kind of just draw the source based on the land use. And then knowing like you've got a couple of neighborhoods that are circled that aren't sewer, that are likely older septic, we can kind of draw a source line there. So I think we could kind of figure out some of that stuff just by knowing what's happening with the land use.
Yeah, that's exactly what I was thinking. Is I was looking at the maps as you guys were going through. Being able to identify a little bit better the individual parcels in those watersheds and sort of what their use was and what sort of was happening, I think would definitely help sort of, I guess, give you that preliminary idea of what the source could be, right? So again, if with the expense with E. coli or the complexity, right, we're not just starting at that point, we're kind of trying to weed out and pick and choose the most likely contributing factors. And I think that I think working with our land use department, planning probably included, like this could be a project that we could probably really help you guys drive down on some of that.
Great. Yeah, and it looks like,
so go ahead, Jenny.
I was going to just say that Daniel Osborne had a comment that might be able to incorporate mapping at the county level and locationality where the septic systems are and how they could influence the results. So he's going to check with the GIS team. So perfect. Thanks, Daniel.
Well, when we can look at having a future meeting and bringing all the, I guess, the local partners together to, you know, see how things can move forward in this direction, at least for Milton.
Yeah. Alan, I see your hands up. I'm sorry, Dana, did you put your hand up before Alan? I'm sorry. Sorry, Alan. Oh, Dana's muted.
I forgot to take mine down before, but I do want to talk a little bit about incorporating data from this or future efforts into the Mercury TMDL plans that are coming for the county and the cities. I know I've already been talking to some of the folks on this call about that, so I don't want to lose that thread. But my other thought on the E. coli was, I was wondering if any of the cities have the ability to quickly analyze for E. coli, analyze water samples for E. coli in some of their public works departments. I know maybe Chris from City of Scappoose, maybe Dave, you can speak to that.
We explored that option when the SWCD established this program, and what they test for and Sarah might be able to explain this better, but basically it's a more of a general test for presence. It doesn't necessarily detect quantity. So yeah, that's limited as to what they can do. So we have to go to outside labs.
Okay, gotcha. But yeah, if we could just talk a little bit about the Mercury piece of it and how maybe some of this could be used in that, or
Dana, I'm gonna put a footnote there in case. Does anybody else have anything they want to talk about E. coli? I feel like we're in a good place with the E. coli, and then we can move on to Mercury. Does that sound okay, Dana?
Okay. Yes, okay. Totally.
Alan, did you want to talk about something totally different? Oh, you're muted now. It's your turn.
Yeah, I would love to talk about E. coli in Upper Beaver Creek, because it's kind of one of those things when I came into the Watershed Council that Upper Beaver Creek was hot. And so it's like, okay, what do we do with that? You know, I'm really interested in partnering with the county and all the partners to figure out a little more granularity of what's going on there. So, you know, what does that look like? And one silly question I have, I think I've asked this before with DEQ, is the source drinking mapped for Upper Beaver or CLATSKANIE? Does anybody know, other than the DEQ data set? Because I did not see a source drinking point for Upper Beaver, just from a public health standpoint. Do you?
Oh, you mean where in the Beaver watershed they have their water pulled for?
Yes.
Oh.
Yes.
I would assume most people are on wells.
Right. That's what I assumed as well. So pretty data poor, other than what you guys have collected. I'm all in for writing grants and getting more. Upper Beaver is, you know, not a salmon bearing creek, but obviously you got public health and watershed health priority for us. So yeah.
do want to clarify that it might be teensy bit confusing. When we talk about the Upper Beaver watershed, our monitoring location is actually on Girt Creek.
Fox Creek, it looked like.
Tributaries of Upper Beaver. So it's not on the stem, it's on Girt Creek, which is a huge sub-basin. But I just wanted to make sure you were aware of that, Alan, because it's very close on the map. You can't tell.
Yeah, there was a map there that I was just looking at that looked, maybe it was probably a mapping error, but it looked like it was actually on Fox Creek for one of your monitoring sites. But that's a silly thing. But yeah, thanks for clarifying. It's Girt Creek. Yep.
Yeah. Jim, did you want to talk about bacteria?
No, I'm good on bacteria and E. coli. I had some other questions about maybe where some of this stuff might lead into the future.
Yeah, maybe it'll lead into mercury monitoring. What do you think, Dan?
I guess my question is more along the lines of, it's not surprising to me that we found these elevated temperatures given where we're looking and the history in these areas in terms of water monitoring. But what really kind of jumped up at me and was somewhat of a surprise was just kind of across the board 1.5 degrees C increase that we're seeing in like Beaver Creek and a lot of the SCAPPOOSE streams. Is there any thought as to why this has occurred in such a short time period? I mean, it's only 6 years, and that's a pretty big jump. I don't know what it could be. I mean, everybody's probably got theories, but have you guys thought about what this could be or how you might influence your investigations moving forward into the future?
Well, I just want to raise my hand real quick and talk about how we were concerned about talking about that. And we really thought about the data, and we really looked into the data and looked into our, we follow very stringent QA/QC processes with monitoring temp data. And historically, I know that we did as well. We had NIST, calibration checks, all those things. And so what we did is we looked beyond the scope of these watersheds, and we found that the Columbia itself has increased that much since that time frame as well. And we actually, in the report, we provided a reference to a Army Corps report talking about temperature increases in the Columbia main stem. And so I think that what we're seeing is potentially climate increasing temperature, air temperatures increasing, stream temperatures increasing throughout the basin. And that, by the basin, I mean the lower Columbia basin. And so, Jim, when we found that data, it made us a little bit more comfortable in our results, although it didn't make me comfortable to talk about how the temperatures are increasing so significantly. That's really scary. Especially, I wanted to throw up a picture of climate for this weekend. And it's like, if we're going to start expecting 111-degree days in June.
Yeah.
Thank you, Sarah. I didn't catch that reference in the report, but that makes me feel a little bit better that it's just not specifically occurring in one watershed. It's kind of a broader extent. And so that's kind of it's a problem, I realize, but it's not unique to one area that we really need to dial in. We just need to follow forward with general BMPs to try to help reduce these temperatures.
Thank you for that question.
Yeah.
Alan, is your hand up from earlier, or you got more stuff?
I got more stuff. Real quick, I just want to dovetail what Jim is saying, because in the CLATSKANIE, and we've started having these conversations about productivity in the CLATSKANIE and why we're seeing so few fish. And one of the hypotheses is that temperature plays a hand in disease like Shasta and Chum and Chinook and all those things. So temperature, yes, is concern for all things, but particularly tied to any sort of disease that's coming on the radar as a result of climate change and salmon population. So, and Jim and I have talked about this. We want to figure out ways to add to what you guys are doing, but also tie it to some of the questions in particular that the CLATSKANIE, I mean, probably it's across all the watersheds about the productivity question of why aren't we seeing more fish? And we really don't know. The productivity question is a major unknown in the CLATSKANIE. It could be a lot of different things, and temperature offers a slight window into that. So just a comment, not a question.
No, and that's a good comment, Alan, and I cannot push for long-term monitoring enough. Like,
Yeah.
Sneha and I are lucky to be involved in monitoring that happens. The status and trend monitoring happens in the estuary. That's Mainston, Columbia data collection. We have a data set that goes back a long time. It's really important because what we see is that the year that you monitor is so critical. And Jim's comment about changes, like what kind of changes are we seeing in temperature? Well, that is important because what years were we monitoring? Well, we know 2011 was a kind of a cold, wet year. And so if we're comparing 2011 to 2015, which was a hot, dry year, we might, what are we interpreting, right? And so the more data we can collect, the better we can understand these systems and understand how these systems are fluctuating, both maybe as a long-term trend of increasing temperatures, but also on an annual basis of just varying. So that's just kind of me trying to sell the long-term data collection. I think it's so important.
Yeah.
Thank you. I just want to quickly thank you for this great effort. This is a great report and a great presentation, and I just appreciate Dana and I laugh that we're in like these poor watersheds, like we don't know what's going on and we don't have the staff time to process it all, but this is what partnerships are all about. So thank you for all of it.
Well, hopefully we can, start. I know Jenny wanted to shout out for her, your guys' like watershed working group. And I think that if you wanted to talk about that now, Jenny, I know you had emailed me about it.
Yeah, Dana and Alan and I just kind of ad hoc started meeting talking about water quality a couple of months ago. And so this was perfect timing that the results came out. And so now I feel like we can deepen that conversation with more voices. So we have our next meeting scheduled July 28th, I think at 9:30 in the morning. And so we can send out an invite to everyone, and you're here today, so you can just go ahead and put that on your calendar. And if you'd like to come and just talk more deeply about E. coli and temperature and all the fun things we're talking about and solutions, that would be great. So we can send out a meeting invite to everybody that's here today. And then I also just wanted to kind of dovetail onto what Jim and Sarah were talking about as far as temperature and kind of our critical species that we're thinking of there, salmon. And when are the salmon using these different watersheds? And what were I think I should look more at the data of what were the exceedances during like spring use periods, because it is pretty common for the summer temps to be so high. And so I want to advocate for us really kind of like prioritizing the different watersheds so we can get down to that focus one that is like, oh, this one's in the most trouble. That's the one that we really need to focus on for temperature. And E. coli, it sounds like we're kind of sorting that out. So it might be nice to overlay the fish use timing and abundance with the temperature issues. So I don't know, Jim, if you have like any like this is the watershed that needs it because of the fish, or if you have that priority at the tip of your tongue or anything like that.
Oh, I think it bears worthy of discussion, but I think I've been talking with Alan and Dana over the last few years about where we might want to be focusing our efforts. And I think right now we should be looking at improving our ability to hold salmon in the upper watersheds and giving them refuge into cold water habitats if they get pushed down. But I think it bears worthy of looking at, maybe some more details in terms of where we think some of our higher densities are and the such, and I'd be willing to do that. But yeah, right now I think looking at this upper SCAPPOOSE, upper Milton, upper CLATSKANIE are all areas that really need restoration and areas where we could start doing a lot of focus. But you can also see papers that have been written recently that talk about some of these middle reaches where the fish are going to be susceptible to kind of elevated water temperature during kind of the shoulder seasons, what they're calling, and they a little bit warmer water temperature is going to help them grow faster. And so you don't want to always overlook everything, but I think you got to kind of pick an area and build out from there. And I'd be happy to engage in those discussions with you guys.
That's good. Thank you. I do want to point out that in the monitoring report, Sneha did write up the number of days exceeding the different thresholds for every month of the year. So it is in the report. You got to read the report. It's going to put you to sleep. I mean, it's amazing.
Hey. Yeah, it's a basic. That's what you say. Yes.
But we're also, of course, happy to answer questions, or if you email us to share that with you. Dana, I see your hand is up. Oh, Celine, did you want to?
Oh, no, I just wanted to get back to, if we're done with discussing the other parameters, get back to Dana's question about the mercury.
Yeah. So another thing that I came up with or wanted to just touch base on is, since we do have a lot of partners, local partners here on the call, can you give us just an idea of what the cost is to continue this monitoring program? And then just to help folks understand that if the OWEB funding does not continue to come through, and everyone agrees that this is useful data to keep moving forward with, what would that look like for these watersheds, maybe at the county level or the city levels, to help you guys with funding to keep this going?
That's a good question. Celine, do you want to talk about it?
I want to say just generally, it costs about $50,000 a year to run this program. And the SWC, OWEB only provides half of that. And the SWCD picks up the other half. And that's just, monitoring for the existing parameters at the existing locations. And so if we were to look to expand the program, yeah, it would, you're looking at beyond $50,000 a year.
Okay. And do you have some idea of, like, per monitoring site, to add one additional site, it costs X, or is it all kind of?
Yeah, I think it would have to be, it depends on the parameters you're monitoring for. Say, like, the bacteria monitoring is more expensive than, a one-time cost of putting in a probe for temperature, that type of thing. So it just depends.
Thank you.
Yeah, and I mean, we do have those costs pretty dialed in. And so, I mean, if you, me, Sneha, Jenny are happy to have those conversations about, expanding or maybe creating a different, like, sub program or whatever. We've been doing this long enough now that we can have it be a little bit modular in terms of thinking about cost. However, I will say that, logistically speaking, those things cost time too. Like, when Sneha and I are doing this, and I should really say when Sneha is out there doing this, because Sneha has been doing the heavy lifting in terms of going out monthly for water quality monitoring since 2019. I mean, how many hours are we in the car?
I'm in the car at least for 8 hours, at least. This is just driving from the office to all the sites and then coming back. So yeah, I start in the morning at 7 a.m. and then by the time I get back to the office, it's 5. So is that 8 or a little more than 8? But basically, that's an entire day of collecting samples, and then on top of that, processing for the turbidity as well.
And she's really fast. So, like, a normal person, it would take 2 days. We used to do it in 2 days, and because Sneha is so quick, we can do it faster now because she's very efficient. But anyway, so adding points and things like that, depending on where they are, it does matter. And so, but at the same time, we have the benefit of been doing this for a while now, so we could think about that differently.
Yeah, I like that modular idea that you're talking about, Sarah, because we designed this to be like a 5-year set of what we're looking for. The questions that we had at the beginning of this period, we're starting to answer them. What is the trend? What is the status of the water quality? And so now I feel like we can ask new questions for the next 5 years, and maybe we're still asking that question again, or maybe we, you know, scale it to say, what are the trends? Let's keep seeing the long-term trends in these focus watersheds, and let's add on some additional parameters for these other watersheds or something. And I'll share the current status of our OWEB grant. We submitted it in April, and we'll find out in October. We did just learn that there were a lot of high dollar submittals for this monitoring grant time period, more than they usually get. So ours is the cheapest of the ones that were submitted for this region, so we are the most efficient. But it is a little uncertain whether that will come through. I see Katie popping up there. Maybe she has some additional information on that. But we're always looking for additional funders for this type of work.
Yeah, thanks, Jenny. And yes, since you mentioned the OWEB funding, I should report that the review team did meet last week, and I'm a little bit late about getting the recommendations out, but the project was recommended for funding. And so I will be sending that out soon. But, you know, it's you guys know the drill. It takes till October till our board meets before they make the final decision. But yes, it was recommended for funding. And I guess I can share that one of the things that came up in the review discussion was that, you know, DEQ and other kinds of water quality folks like to see, you know, at least 8 years of baseline data. And I know the application mentions this as the 5th and final year, but it definitely sounds like that the technical experts are really interested in seeing this continue, you know, past the 5 years. And so I think the discussion that you're having right now is really pertinent about where you would be looking, you know, after this. So, yeah.
Yeah, like, of course, thanks so much, Katie. And like the value of a long-term data set is always like, you can't put a price on that value. And of course, like 8 years is much better than 5 years, because on like within the first 3 years, or like between 2017 and 2020, we're just starting to form these trends. So definitely, like, we can, you know, point out more focused results and more variability. We can reduce the variability in our results as well if the program goes further than 5 years. Yes, absolutely. Thanks a lot.
Yeah, thank you for that update, Katie. Now, Alan, your hand's been up for a long time. I'm sorry. I just noticed.
No, it just came up again. I'm good. This is my third. I don't know how many hands I get to have. It feels like just not another observation that like the first 2 years, and we have some data in the you know, a decade ago, and we can do more intensive studies. It just feels like the time is now more than ever to engage in landowners before, and this gets submitted to DEQ and all the things. This is a perfect role for the watershed councils to perform that soft social work. We have the data in the back of our pocket, engaging landowners so that it doesn't get into, you know, a regulatory thing. And then we lose our partnerships, we lose our trust and relationships, and it just feels like. And I know there's, you know, there's stakeholder engagement grants out there, but there's a lot more information now. Maybe we should have done, you know, obviously we should have done this 10 years ago, but, you know, anyway, as far as the Watershed Council role is concerned, and I think Dana would echo this, that we don't need to do another study to engage the landowners. We should be engaging them like sooner rather than later, because so we don't lose that trust in those. We need to build those trust and relationships now before, you know, things get, you know, it's Columbia County, before things get curious with landowners. So that's just an observation more than anything.
And I know that we, as people who are not engaging, you know, me and Sneha are on the ground doing the data collection, but we're not engaging the landowners unless they come and talk to us. And while doing the monitoring, which can sometimes be seen. But I do think that you're 100% on target. And if anybody has a contact or a landowner and they want to talk about the data and they want to engage Sneha and I in a conversation, like, we are very open to that and happy to fill that role and be very diplomatic. You know, it's just not something that we are doing actively, because that's really the role of the watershed councils and the Columbia County Soil and Water Conservation District. That's been, and I am very, I want us to be friends with landowners and come with productive solutions that make everybody happy, that are sustainable land use and water quality. So I'm there with you, and I'm happy to troubleshoot those. issues. And I think it's really it's the elephant in the room, Alan.
It's the social work, no big deal. It's never ending. But yeah, thank you. That's great. I just, I'm interested. There might be a stakeholder involvement grant coming your way because we have more information. I think we have more targeted. We have more focus now in these areas, a lot more information. I didn't mean to say we were, data poor or anything, but we have enough information moving forward to be like a little more strategic in our involvement and target our efforts. So.
Yeah, I just want to add, kind of tying it back to the mercury stuff. Part of the requirements of those implementation plans for the city and the county is to do education for landowners about preventing erosion getting into streams, but it also just ties into stormwater management, buffering the streams for, general water quality that I think, could really result in a robust education program that either the Watershed Council, the Conservation District could take the lead on, because I don't see the county and the cities having not the capacity, but just the relationships that maybe the district and the Watershed Council have built with, being able to offer that sort of education in a non-regulatory way. So maybe that's one way that we can start building towards a landowner education and outreach effort that would target a lot of these different issues with one effort. So I'm hoping that in conversations with the people developing those plans that we can, maybe Celine, you and I can talk about how we can incorporate some of that into that to kind of meet all of these goals, as well as, a monitoring effort that's going to be required of the cities and the counties to show progress on reducing total suspended solids, turbidity. They're going to need to do that as part of these plans. So maybe that's a way they could also add some more long-term sites or some sites that they can use to also capture that data. So that's kind of where I think the mercury and this water quality could kind of overlap, is now they kind of have to do it. It's kind of something that has to be done. So why not wrap it into one effort instead of trying to duplicate efforts across these issues?
Yeah, I think that's a good point, Dana. And I really think that, buffering these water courses is, and that is something that a conversation that should be started at the planning level too. In Washington, there are mandated buffers off of water bodies. And I know that in Oregon, depending on the jurisdiction that you're in, there can be mandated buffers as well. And I believe that some of the jurisdictions in Columbia County have some mandated buffers, but they're pretty narrow. And so I think that it would be really constructive to pull in some planning folks and start talking about buffering. And then that's something also that, the SWCD could help in, as far as this element, but planting. We need more substantial vegetation other than reed canary grass along many of these waterways. So
I think therein lies a big challenge that we've talked about with Jim too, is, we can go in and try to plant these lower watersheds all day long, but is it really where we need to concentrate our effort and grant dollars? Or, do you concentrate on the upper watershed and just leave the lower watershed to the landowners to try to take on the cost and maintenance of that themselves? Because man, it's a multi-year effort to get a tiny piece of stream in the lower watershed cleared of reed canary and blackberry and planted with something that's going to survive. So it's definitely an ongoing conversation.
I hear you, but stormwater regs at a state and federal level are probably going to, particularly as it pertains to mercury, because of the fish issue within the endangered species within the Columbia River, is down the pike. So these jurisdictions can either start wrapping their heads around that they're going to get regulations placed on them to have these water quality parameters fall within certain metrics. And they're going to have to incorporate some things into their planning processes. And so maybe that is something that can, because understandably watershed councils and even OWEB, they can only do so much. And also the goals are a bit different.
Right.
So, but just starting the conversation would be at least a starter. So
I kind of want to add in on that if I can real quick, because our planning manager, Matt Laird, and I are just talking about this. Like literally just yesterday, we were talking about mercury TMDLs, our responsibility that we think we're going to be having to do serious upgrades to our erosion control plans, how we spread that out, what that looks like, talking about riparian areas and our ability to enforce that. And when I say enforce, like I want people to know that our model at the county is very much, we try to work with people and educate people and that sort of thing. Ultimately, yes, we are regulatory. But I agree with what was said that like this conversation with our planning staff should be happening right now because it's on their radar and because they're just going to be starting this effort, trying to wrap some of this other stuff in. Like this is the perfect time to be talking about these things and being a little bit more comprehensive in the effort rather than just, oh, we're being told we have to look at mercury TMDLs. Well, what else can we sort of build into this to kill many birds with one stone sort of idea? And then just to tack on a little bit with like the previous conversation with education and reaching out and, social work and whose responsibility that is. You're correct that probably should be coming from Watershed Council and Soil and Water Conservation District. But I know the county, because I've advocated for this many times, like we are not opposed and have the ability to share information, right? To like promote information that you guys are putting out there and saying, hey, this is important. This is the message we're trying to send. Like, those things can be included in processes. And I think that we're all willing to incorporate that where it makes sense and where we can.
Cool. That's great to hear, Erin. I would love if you could join our little water quality, you or Matt, or someone from the county. I've been briefly chatting with a few folks, but just to kind of, keep this conversation going locally about what the next steps are and how we can get it all together. So
yeah, I'll talk with Matt tomorrow about this meeting. And then you guys were saying that you were going to send out the meeting invite for this idea that you're already talking about.
Mm-hmm.
I missed the date and time, but obviously I'll keep an eye out for the meeting invite and, I can sort of loop him in on that too. And, ultimately it's up to him, but literally he was just talking about it. So I think he would be interested.
Great.
Well, thank you guys. That was really informative. I was not that familiar about the mercury TMDL work that's going on. And I think that Sneha and I are both going to start really looking into it now. I'm not as sure exactly what the protocols are for grabbing those mercury samples. And so that would be really interesting to learn more about because that would be helpful to estimate cost and things like that. I'm sure that's stuff everybody's thinking about.
Interestingly, they have a proxy of total suspended solids for mercury. So I was curious if you could use your proxy of turbidity as a proxy for total suspended solids as a proxy for mercury, but I'm not sure if there's a 3-step thing, but I know you can test total suspended solids and get a mercury estimate. So I think that would definitely be one that folks around here are going to be interested in adding in some form or another. I know City of Scappoose was looking at, can we grab a sample when it comes into town, where South Scappoose comes into town and where it comes out, so they can essentially show some improvement. At the county level, it'll be a little bit trickier to figure out where to focus that, where would be useful to show improvement. But I think something similar in the city of St. Helens would also be helpful, sort of at the edge of their jurisdiction and the end of their jurisdiction, getting some some sampling done, but I think we can kind of get to that through some of these water quality meetings that we're talking about having. So
yeah, I will say that I know that for total suspended solids, a lab that processes the bacteria samples will process a total suspended solid sample. So any place that we collect a bacteria sample, which is all of them, we could easily add a second water sample that could be tested for total suspended solids. I just don't know what the cost would be at the lab level, but it does sound like if it's something that is going to be a widespread test, it would be worthwhile to think about adding. And that kind of thing would not, time-wise, it would just be a matter of us grabbing a sample while we're already out. So I want to be cognizant of everyone's time because it is 4 o'clock. And Aaron, I see your hand is maybe just up residually. Okay, I just want to make sure. Sorry, it took a long time. It was a really robust conversation. I didn't want to interrupt, but it seemed like it flowed right into what you were talking about, Aaron. Sounds like we're going to have a follow-up. Either we could follow up with the Watershed Working Group, or Celine. In addition, we should probably have a meeting. Celine talked about maybe having a meeting with Aaron and talking about the bacteria data. So, kind of thinking about following up on this. I really appreciate everyone's time today. I felt like it was a really robust conversation. If you have follow-up questions, please don't hesitate to email any of us. And we will be posting this for public view. This is the video that we have taken. And so I think Sneha can go ahead and end the video now. So thank you for everyone. We're not ending the meeting, but I was going to end the recording.